is that there's instead they assume that if altering some dimension depth of pit, width of aperture, etc., leads to some selective advantage to increased acuity. They say the angular resolution, increased angular resolution, then selection leads to a 1% change in that dimension per generation. So this is math bio. What's your background?
Math and econ.
Math and econ, right? Okay. I mean, so mathematical biology. Actually, it's not even mathematical biology. It's theoretical biology. One of my colleagues, who's a theoretical biologist, says that even the theoretical biologists admit this is cloud cuckoo land. Your models do not have to make contact with reality in the same way that is required in computer science, where you've actually got to simulate something. If you propose an algorithm, generally you've got to implement it. If you propose a model or learning system, you've got to apply it to some data. If you propose a clustering method, you pick a standard data set and do it. You can fix that, obviously. As you get towards publications, you will discover in your projects, but you've got to do something. This is an analysis. So they've made this very interesting assumption. I think it's a very interesting assumption. They say they identify there's some measurement. They'll say that selection can change it by 1% generation. What has to happen for that to occur? For something to change by 1%? Well, what's the assumption here?
Some mutation.
Well, yeah, there's a more basic assumption.
That like everybody changes it at once?
Everybody changes at once. Well, they say they defend it by saying, okay, the reason they can include a pessimistic estimate in their title is they're assuming that they're only going to change one dimension at a time. Okay? And they're saying you can change it by 1% change. So they're assuming that there is variation in the population. And supposing you're trying to deepen that optic pit, you've just got a little light sensitive patch, and you want to deepen that pit, they're assuming that if you take your little sort of swimming eyes or whatever, and you separate all ones with the deep pits, and you kind of squash the rest of them. No, you're a humane person. You put them into an alternative breeding line, a control experiment. You just squash them. Then you're assuming that the children of those little swimming eyes with deep pits are also going to have deep pits. That's quite an assumption. Might not be true. Now you say, hang on a moment, yeah, but children are like their parents. Well, yeah, but why? So they're making let me put up another. There's a sort of hidden. I'm going to use orange. There's a representational assumption here. There's a that in genetic representation, whatever it is, they never talk about how it might actually be represented in genes, is such that children resemble parents. value of this dimension. And there will be variation. Like genetic variation. So it's assuming that there's going to be enough mutation in that, enough mutations around, or variations in whatever they're interested in, or whatever dimension they're interested in, to be present. Then they're going to select away, and they're assuming that if you select the ones with the deeper pits, then the children are going to have deeper pits. And nothing else is going to change. That's quite an assumption. You think, wow, what an interesting genetic code that is. I mean, one of the extraordinary things about genetic code is that if you take your mother's genetic code, perfectly viable mother, father's genetic code, perfectly viable father, their genomes, and you sort of snip them up and recombine them in some way, in a pretty random way. That is, the corresponding parts connected to corresponding parts. The chances are very high you get a viable individual. Well, try doing that with an HP laptop and a Dell. Does not work. Try doing it with 2 suspension bridges. Very unfortunate. So the representation, whatever it is extraordinary, and they're assuming, without even saying so, this property of the representation.
Could you, sorry.
Yeah.
Is it possible to get around that assumption by assuming that the property confers a higher survivability? It may not be explicitly discussed here, but
yeah, okay. The question is, can you get around this by just saying that the deeper pit is really good, right? Yeah, okay. There is this notion called of the use of heritability. I go more deeply into this later, but basically, let's put this. Say let's take this as the mean of the parents, mean of the parents' measurement, whatever it is, deep depth of pit. So yeah, let's measure this. Take lots of these lies, go and breed, just get children lies and measure them. Okay, measure the parents, measure the children. This is the child's measurement. It could be height, it could be. And when you're hoping, so there's going to be variation. It's going to be. So this will be unfortunate, right? A scale like that is really unfortunate. You select the best parents, damn, the children are all the same size. Is that small parents? So you're hoping that there's going to be a scatter plot where it's going to be something like this. And you put a line through it like that. And I think they make, so what assumption about heritability? What's their heritability figure they take? They don't, I mean, typical figures. So now, the amazing thing is that for pretty much anything, you'll get a heritability of sort of 0.1, 0.2, 0.3, 0.4, 0.5. The regression line for height with humans is something like 0.8. If you find a trait which is not some measurement which is not inheritable, wow, you can publish a paper about it. If you breed, you take pretty much any animal, and you have a breeding population of 50 to 100 animals. The classic experiment was flies, and the thing that they decided to breed was they took the fifth, you turn a fruit fly over onto its back and you stick it under the microscope. It's got little plates down its chest. And you took the fifth plate and they count, and when you look at it under the microscope, it's got little hairs on these little plates. So they counted the number of hairs on these plates. Usually they're about, I can't remember. You breed the fruit flies, and each generation you count the hairs. Graduate students had a great time doing this. Okay. This experiment was actually done. Thousands and thousands and thousands of fruit flies. Yes, you can breed and you can increase the number of hairs. Any property you like, you can do this. And so generally, the representation is such that not only can you chop two genomes up, put them together, and you get a viable individual, but also the measurements you take with the children are correlated. The children are to some extent correlated with their parents. So they're assuming this. And this is such a natural biological assumption that it's sort of slide it past you. All the vegetables you eat, every single vegetable you eat is descended from some wild plant. The vegetable you have looks nothing like the wild plant. This has been done partly through accidental breeding, and the people kept the good ones and tended to sow them, and for the last 50 years by deliberate breeding. So there is a large industry of breeding animals and plants to increase characteristics. One statistic I think is, you know those black and white cows, those Holstein cows in America, they're always in the cartoons, American cartoon cows, with black and white patches on them, right? Those cows, I think, this was around in the 1940s, one mother cow could provide enough milk to feed 3 calves. And after 50 years of breeding, selective breeding, the cows you see in the fields, if you drive around and you see dairy cattle in the fields, or certainly on large industrialized farms, particularly farms in America, these cows are created by artificial breeding from semen from bulls with special stud farms, and those have been selected so that they have to be the ones that have the daughter cows that gave the most milk. There's a huge industrial process. They're usually selecting for about 15 different things at once. And after 50 years of this industrial process of selection, one Holstein cow, 20 years ago now, gave enough milk to feed 7 calves instead of 3 calves. It's kind of changed people produce. These quantitative changes. Yes. Unless you're eating a rare breed, in which case you're dealing with the accidental and amateur results of selection in previous centuries, you are eating food that has been industrially bred for selection now. So it's something's done like, hang on a minute, I don't care if my food's organic, I think I want it to be a rare breed. And this is a whole other topic, but it isn't really. It's another very remarkable thing about genetic representations. Which I'd love to be able to explain. The truly, I'll tell you the truly remarkable thing. Supposing that, I can't remember if I've even got a slide on it. If you imagine, this is an experiment that was actually done. In 1890 or so, they decided to check out in Illinois, in the States. They decided to start an experiment of breeding maize, corn. And they decided to sow a field of corn, and they'd collect the kernels, and they would measure the oil content, and then they would select out those seeds which had the highest oil content, and those seeds which had the lowest oil content. Then they'd take those 2 lines of corn, sow them in the fields again the next year, and do this. They were breeding 2 lines, one for high oil content and one for low oil content. I'm sure I probably have the graph somewhere. The low oil content started off with 5%. As you can see, over 70 generations or so, the low oil content went from 5% to below 1%. The high oil content, I can't remember the figures, 20%, 30%. Massive changes. And the point is that even after a few, even just 10 generations of selective breeding, on pretty much any measurement you like, pretty much any animal you like, you will find individuals who are way outside the wild. So if you start off, imagine you have your measurement here, whatever it is, height, oil content, or whatever. So if you plot a little histogram, this is your initial population. If you plot a little histogram, you might get this. Okay, and so you have a sort of a mean, and you have a sigma here. Okay, so this would be your initial type, your wild type, as the biologists call it. Then it is typical that after a few tens of generations of breeding, you get to, over here, you get various things happen. You get a wider, this isn't even extreme, this is, I don't know, the mean here is 5 sigma, 10 sigma, 15 sigma away from the wild type mean. So you can change pretty much anything by selective breeding. So Nilsson and Pellegrin basically assumed this result. Probably, in that. But it's a remarkable thing. What else have they done? What else do you think about this paper? Yeah, they didn't program. They've assumed certain miraculous things, which do indeed happen in biology. What else do you think about it?
Can I just go back to this? Because then this is, you get these very high standard deviation results due to selective, some person selecting it.
That's right.
Then in this case where there isn't someone selecting it and it's up to nature and other constraints to do it, is it an acceptable assumption?
What they have made an assumption is that they said all we're going to assume is that we assume that during the evolution of the eye, that individuals that have eyes with better angular resolution were more likely to reproduce than individuals that did not have such eyes.
Oh, okay.
Okay, so the better your angular resolution, the more likely you were to reproduce. There's a small selective advantage, which is kind of plausible. What do you want an eye for? As they point, and so they're purely interested in the optical properties of your eye patch and pit. Yeah.
But although they use the eye as the indicator of evolution, at the end they have an interesting analysis that the eye requires a system to support it.
Very good. So yeah, absolutely. Yeah,
the worm with a massive eye becomes a fish.
Right.
Right.
Yes.
And the fish is a fish only if the environment can feed the fish.
Right.
So there's like a network dependency that somehow is not there.
Absolutely.
That could blow up the whole eye indicator, but it's interesting just as a first tryout.
Exactly. The point is that if you've got your wonderful eye with your graded refractive index and length, you've got to have a brain, and that brain's got to be involved as well. And there's not much point in having. There's not much point in a limpet that just sticks to a rock and sits there and doesn't actually do anything. It's only going to have a selective advantage for really great vision if it's going to use it in some way. I mean, if you're a little, there's little things, jelly-like things that stick to the rock. You have a fantastic eye, and you sit there and you see this fish, and you think, oh no, I can see this fish. I can see it from 20 feet away, and it's coming towards me, and it's going to eat me. You've got to be able to escape. There's no point. So exactly, there are all these multiple dependencies of various kinds. You might also want some muscles to move around. Yeah.
The suggestion that the reproductive, the probability of reproducing would be higher with this eye modification. I guess that would bring us to this problem that you were discussing earlier, that these properties aren't necessarily inherited in the following generation. But is it possible to swap that assumption, because it seems a bit less realistic than saying survivability if you can see predators near you, or you can more efficiently search for food, so that survivability increases. So replacing that around, does that not take care of the issue of passing on the traits in a sort of passive sense, not because it's genetically passed on, but because those that do have that trait are more likely to survive, so that trait continues on to the next generation.
Okay, well let's imagine we have these little eyes wandering around, they've got those little tadpole-like tails, and then they face that great test. The sharks come past, and these eyes, they use that eyes for just sort of dodging the sharks. And the sharks are kind of migrating, and they'll eat some of these eyes, and the eyes that don't spot the sharks and don't dodge get eaten. So the ones that survive are going to have better vision. They're going to say, phew, God, the sharks have gone, okay, let's breed. And they'll breed. So at this point, you've got some acute eyes. Then they lay their eggs, and they die, and the eggs hatch, they're their children. Now, if the children, for this process to continue, generation after generation, to produce continuous change, the children have got to resemble the parents.
But they need not resemble them to as great an extent. Only
that's right. The slope of this can be like 0.1. Yeah, it can be quite slow. Even if the slope is pretty low, if you have a lot of generations, then and a large population, then you'll get change. That's true. So, but provided you have to have some positive correlation with resemblance.
Yeah, but the impact of the environment is also key. Because let's say there was a change in the environment, there's no light now, but there's an outlier who has a modified eye who works with ultrasound.
Yeah.
And the ultrasound will be, the few 5 ultrasounds in a 1,000 population will be the only ones who can pass that genetic information to the genetic simulation. So somehow they did not include a vector of the pressure of the environment.
Yeah, I think it comes down to that they didn't have a program. Because if they tried to build it so that the evolution went forward, they might discover all sorts of bugs in the process. You have all these blind alleys. You might get sort of pits in such a way that you'd never get a lens. You might have multi-pits. It might be easier to evolve. They're taking the path that they know, and they turn it around a bit, but at the end of the paper they say, actually, the plausibility of our account is really shown by the fact that there are organisms out there who've got eyes really at every stage of this development. And you think, yeah, well, you knew that before you wrote the paper. I mean, that's why you chose this upgrade path, because you knew there are light-sensitive patches, there are pits, and all these things. Okay, so it's again this problem. They're doing a backwards analysis, not a forward simulation. I think it's another part of that problem. I mean, as you say, there's the ecology as well. All those problems. It just doesn't even occur to them to do a forward simulation. I mean, there's no way they're going to try and do that. Which I would argue is a huge weakness of this thing. I mean, it's an achievement. They've got an analysis, they show there's an upgrade path. What else? Did any of you look at the second paper? The other thing I'd like you to know about this paper is the degree of polishing they did to get their analysis elegant. You know, they were considering all sorts of. I mean, you know, they spent months getting this into a nice mathematical form. That's mathematical biology. Now, what about the second paper? The second paper, did any of you get a message from this? Yeah, it's horrible to read. I find molecular biology papers really hard. Yeah.
There was something very interesting. The multiplicity of paths. There's not only one way that how it can happen, and how the molecules can be indicators of the path.
Yes.
And how they recombine, appear, disappear.
Right.
So it allows us to see a wider variability in the process.
Yeah, the second. Can I hold up a copy of the second paper to show the the second paper? I think there's a. I mean, it's much harder to read because they're saying it could be this and it could be that and it could be that. And about every single case they're describing, they have imperfect knowledge, and they don't know quite what's going on. They have any indirect evidence on this. For instance, it's this diagram here. So this is an evolutionary view of lots of things in plants. And it starts with sponges at the bottom, and it has the earliest, the sort of 600 million years ago, bifurcation in animals was between animals with circular symmetry, they just have a top and a bottom, and what they call the bilateria, the ones which are sort of, they have a left and a right, and indeed a top and a bottom. And so they show that they then give a diagram of various types of eye, rather crudely, and they find that some of the same signaling proteins are critically needed in development of all of these kinds of eye. But at the same time, there are interesting differences, as you point out, in all of these cases. So that there are a range of proteins. There's PAX6 and PAX2, and the point is that whether a protein is being made or not by a cell depends on what other proteins are present, because they stick to the DNA and regulate it. And so there can be mutations which proteins can be captured. If you like regulatory capture, there can be a mutation that causes it to be regulated by one more thing, which changes the pattern of regulation. So that there is gradual change of these regulatory patterns and developmental pathways. But it's this paper gives you really good evidence, mainly because of this single thing, PAX6, that actually the developmental path for all eyes, the squids and the octopuses, which I mentioned before, our eyes, and sponges and jellyfish, are actually descended from the same ancestor. Because otherwise, why would they be using the same protein? There's nothing so special about PAX6 that connects it to eyes. It's just one among many regulatory proteins. It's a pretty flexible one. It can be regulated in various ways. So, what I find astonishing about this is that there weren't. Actually, eyes are all descended from one common ancestor. And yes, many structural developments took place, but there was really only one development of eyes, which is a rather different picture from what you might get from the Nielsen and Alpelga paper, which sort of seems to indicate that eyes could develop within 200,000 years. In principle, any organism gets a light-sensitive patch can develop an eye. Actually, this process happened to different degrees only once. There's one further thing, and I think you mentioned ultrasound, which I rather like. I teach a course on digital sound and music, and sometimes I go through the whole course and nobody asks me an interesting question. It's one of those times that somebody asked me an interesting question. And he said, well, why don't we have as good hearing as bats? why can't we do echolocation with ultrasound? Well, that's a very good question, because I mean, wait a minute, let's just think about this. All those bat ears are tiny, but bats are tiny little things, they're about that big and they fly around. And you know, we were sort of camping out on the dangerous savannah of Africa, with kind of surrounded by big cats that can see better in the dark than we can. Hang on, why's that? Why can they see better in the dark than we can? We need to avoid them. We were wondering, there was no electric light. This is close to the equator, you have 12 hours of darkness. And when there isn't a moon, it's really dark. You're wandering around, you sort of bump your head into trees and stuff. You kind of sleep, it's difficult. Echolocation like a bat would be really useful. Our brains are absolutely enormous. Like a bat's brain is only about that big. People have brain injuries and it doesn't need to affect them very much, where you lose much more brain matter. We just fit an entire bat's brain into one of those cavities, have a little bat identity. Okay, we have little funny noses with little flowers and stuff. We'd think they were nice. And you go sort of squeak, squeak, squeak. And after dark in the savannah, wouldn't it have been great to go on sort of squeak, 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 and you just see just as well as by day with ultrasound? That'd be fantastic. Why don't we have that? But again, this assumption slides past that, oh, no, it wasn't so useful for us, so we don't have it, we lost it. Well, it would have been a bit useful. So, to what extent are we up against the informational limits of complexity of evolution? It's a very interesting question. And again, in this Nilsson and Pelger paper, the first paper, yeah.
Sorry, there are blind people I've seen in the States that have taught themselves how to echo. Okay, not with ultrasound, but just by clicking there.
Absolutely. I mean, we can. So we do have this ability to a certain extent. But I mean, if we could go click, 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 like sort of bats, why couldn't we? I mean, I guess we need a clicker. Then, if blind people could do it, well, we were blind for nearly half of every 24-hour period. Wouldn't it have been useful?
But there's a biological trait that we generally don't know, is that the amount of neurons that we have in the brain related to hearing sometimes is more than to the visual senses. Right. So somehow, or we don't know it, the hearing could be more relevant for everyday information system than the eye. So somehow we don't see it, but it's there. And on the other side, the people who echolocate, they observe that they develop their brains as if they had optical devices. So the neurons that see are the ones that are developing those brains.
Yes, I agree. Everything says is right. And if you're blind, then you divert neurons to parts of your cortex start processing sound rather than vision. The trouble is the bat's brain is still only that big. Yeah.
Well, another implicit assumption here is that people are sort of living in the wild and therefore exposed to these dangers, and therefore that selection pressure existed. And I think there's a severe discounting of the fact that these societies, whilst nomadic, were actually very well organized, and that would have reduced that sort of selection pressure.
Sure.
And clearly there was no need, because people were sufficiently organized and structured in their living to not to need that.
But you're also assuming that we can just, if there was some evolutionary pressure, that we can just switch states. It could be possible to host species die off, right? You come down this path where you can evolve into echolocation or something. That could be possible too.
I mean, it's a slightly sort of. I mean, I think it's a reason. So maybe there's a conferred no advantage because, but I find it hard to believe that something nearly as good as vision with ultrasound in total darkness wouldn't have. Hey, who knows? Maybe it wouldn't. Yeah, I agree. Again, Nilsson and Pelger assume that they're both assume that they're making out that the evolution guy is easy, and they're also making out that you don't have to. You may not get all the way. But eyes may be perfectly, simple eyes may be perfectly adequate for. Why I'm a little bit uncomfortable with this assumption. That there seems to be a kind of, put it this way, you may need to use, let's put the question another way. What if each individual only generates a certain number of children, there is only a certain, if you like, selective ratio you can tolerate. You don't have a 1 in 1 million selective ratio. So how many things can you select on at once? There's a certain selective budget. So what you're going to, so in a sense, you can regard a species as spending its selective budget on maintaining certain types of structures against mutation. So the question is, what is the selective budget? Well, again, I think it's something that Wilson and Pelliver don't consider quantitative. With the second paper, I think it's maybe there are kind of massive coincidences in evolution. Would it be possible to find a planet on which organisms, how probable is it that eyes would develop at all? I mean, the huge variety of eyes developed out of one event, it seems. So there are a number of kind of questions. It's not terribly complex. So what I want to do is let's come up with a really simple genetic algorithm to study. Let's try that. Okay. And let's take yet more looking for let's begin to do this. It is warming up, jolly good. Let's go down here. Let's take a tour of life and see. We'll do better. Doesn't want to. Oh, good. Right. Let's have a look at some. Point to other sort of curious facts, a few of which, many of which you look at from a computational point of view, and you say how. There are kind of three worlds of life. There are what are known as eukaryotes, which is complex organisms. Anything you can see is usually a eukaryote. Eukaryote means, Greek is good chromosomes. What it means is it's got chromosomes rather than loops, and essentially it reproduces mostly sexually. These are protozoans, animals, and plants, all complex organisms. Prokaryotes are essentially bacteria. There are two kinds of bacteria. If we lived in a microscopic world, the main variety we would see would be in bacteria. Bacteria would be as different as fungi from elephants. And there are also viruses. There are kind of, to an order of magnitude, there are kind of comparable to a few orders, a couple orders of DNA of genetic material in viruses, bacteria, and eukaryotes. Eukaryotes are bigger, we've got lots of cells, we've got lots of DNA, but there are lots of so in all, there are three classes of life. Viruses again have enormous variety. Some viruses, such as HIV, actually duplicate from two strands, swapping between them, so they have recombination, so that they actually have a sort of quasi-sexual reproduction inside a cell, which is may make them evolve faster. Luckily, they don't have very much junk, so their genomes are still very short, which reduces the advantage of that. And I'm going to be mostly interested in these, in the central ones. Just to say again, if you were microscopic, you would see a gigantic difference between the prokaryotes and the eukaryotes, and the difference is that the cells of the eukaryotes look completely different from bacteria. They have utterly different structure. They're much bigger. Their genomes are much larger. They contain little kind of vestigial bacteria, which are symbionts. That vast numbers of this is a eukaryotic cell. The size difference doesn't. This is hundreds of times larger than that. Any small bacteria can kind of live inside your cells. The reason why tuberculosis is so hard to get rid of is that TB bacteria actually can live inside your cells. So it's kind of hard to zap them with correct antibiotics. And this has a finely divided structure with multiple compartments, and as well as bacteria are of many different types, but that DNA tends to float loose inside a single compartment. While others are trying to look at, so it's kind of bizarre that there are sort of three totally different kinds. There is this huge variety of life, all existing simultaneously. Well, one bit of it wiped the rest out. How much DNA? Sort of 30 or 40 years ago, I was said, right, okay, well we can, so that some things are more complicated than others by how much DNA they have. And they discovered that you couldn't. This is the amount of DNA in a cell, and you discover that the simplest bacteria, the smallest bacteria, have about have a few 100,000 bases, I think, in their. This is in picograms. It'd be better if they did it in terms of bases. But for other things, there's a logarithmic scale, so you can see the amount of DNA in a plant cell can vary by a factor of about 1,000. That is bizarre. Some plants have 1,000 times as much DNA in their cells as others. Amphibians have similar problems. Insects, where are the mammals? Mammals, mammals, well, okay, they may not have as much DNA in us as rice or wheat, but actually the reason why mammals are so restrictive here is that actually the mammals are relatively, there aren't very many of them. About 10,000 species of mammals. There are far more species of millions of species of mollusks and insects. Millions of species of insects and mollusks. There's much larger orders. Mammals are tiny. Reptiles, crustaceans, sea urchins. Like some sea urchins have got as much DNA as we have. So it clearly doesn't go with the amount of DNA. You've probably seen this before. Minimum genome sizes. At last, they wanted to have a graph, a plot, that showed that people were complicated, and they found that the minimum genome size you need for a mammal is bigger than for other things. It's quite a satisfying graph. What I'm getting at is that biology is messy. There are it's a little bit like neuroscience. Are you studying any neuroscientists here? You've seen neuroscience textbooks. I would have brought one in to show you, but it's kind of hard to carry. Neuroscience textbooks are so big, they're especially large paper, they're especially thick. They have 50 chapters. The reason is we don't really know what's important. We study everything. So we have very little idea of what actually the brain circuits actually compute. And so what do you leave out? Because they didn't really know. Do neurons do... well, I'm not going to go into neuroscience. But there's a problem. In terms of evolution, there is an enormously complicated, messy process happening. What simple model should we choose? It's really quite hard. We want to call something clean and simple. We can mathematically analyze the model and maybe apply. And it's easier to generalize and apply to practical problems. So, what can we do? What is necessary for evolution of complex organisms? Actually, there's a beautiful book called which I thoroughly recommend if you like reading, if you like high-level biology, I thoroughly recommend The Major Transitions in Evolution by, I think it's Zathmary, Hurst, Zathmary, and Maynard Smith. Now, these are eminent people. This is an intelligent book, and it's an overview, and I think it may have the 5. They identify 5 major transitions in evolution. So it's an attempt at a grand picture. Whether they're right, if there's other major transitions, I'm not sure, but they're intelligent people and they want to do it. So, what can we say? First is that really, nearly, well, insofar as you can ever say all in biology, you can say that all complicated multicellular organisms are sexual. They reproduce sexually. And there is no significant asexual lineage of large organisms. You need to qualify this a little bit. There are plenty of plants that reproduce asexually in various ways, and they can either kind of fertilize themselves, so they can kind of commit self-incest, or they can just kind of propagate by vegetative propagation, and so on. But essentially, these lineages tend to die out. Asexual reproduction, also known as parthenogenesis, in large organisms evolves repeatedly, and those organisms tend not to last. You shouldn't sneeze at asexual reproduction. There's one plant that reproduced asexually by just kind of vegetating and budding, and it invaded the whole of Western Europe. So you can go on. But bananas are asexual, for example, at the moment. But the problem with asexual reproduction is that you get very little genetic variety and much slower evolution. So it is believed, biologists are reasonably sure, or theoretical biologists strongly believe, that sexual reproduction is better capable of producing complex organisms than asexual reproduction is. There is an alternative explanation, which is that sexual lineages form new species, can split into different species faster than asexual lineages. So in the evolutionary tree, if you have bifurcation of sexual organisms, which is faster than bifurcation of asexual organisms, then your tree is going to be wholly sexual, meaning that the asexual organisms are wonderful. I'm not sure I really believe that. The same thing is the amount of DNA. Junk is actually a somewhat unfashionable word now, since they've discovered continuously more and more functions of DNA that was originally believed to be junk. But there's certainly very large, I should really have written here, very large genomes. Again, it's not clear. There's a reason it's not clear, is that if you have sexual reproduction, you're likely to spontaneously generate a large genome by the following mechanism. Again, and what I'm trying to emphasize is really how little is clear and how little is certainly known. Let me draw you a crossover event with sort of a green chromosome. So a parent has 2 chromosomes, and you have a crossover event. I mean, I assume I can talk about crossover. I'm not going to explain that, otherwise I could do a test and see how many people went to sleep. But I don't really want to do that. And if I went to sleep, it would be very embarrassing. So the problem is that how crossover actually works in nature is that in genetic algorithms, your genome is characteristically an array of length, conveniently n. I'm going to use L. So you just choose an index where you're going to have a crossover. Simple, bang, program does it. In nature, the genome is not an array, it's a string. So you've got to find a place how does the cell decide where it can validly have crossover? And what happens is that they tend to kind of conjugate, these genomes kind of conjugate, and then it goes together here in the complex forms, and these sequences need to be the same. Or I forget, it's like 20 to 30 bases. An awful lot of crossover events don't quite work, so that actually you get one sequence copied into the other. But they copy up, and then they sort of snip out and they cross over. But so they identify where to cross over partly by general location, but also partly by identical sequence. But what's the problem with that? What bug or feature will happen? How do you get a bug?
You always retain that feature if it's always been copied.
If you're identifying where to do crossover, okay, so this goes to.
It could be at any point on the genome, so it could be different lengths on even parts of them.
Yeah, exactly. So the good version is this, and then you've got this, and these go off into your two different gametes, right? So this is tick. This is your, in software engineering terms, I think you call it the happy day path, right? Or normal use case of your software engineering. This happens there. This is good crossover. Done it right. What can go wrong? What can go horribly wrong? They join together because the two sequences at this point are the same. What can happen?
If you cross over at the end of one and the other end at the other, you could end up with something almost twice as long as the original.
You could. So what happens is if you have repeated sequences, which you do, yeah. So but, and let's do this in red, but it's just so we can use all these at the same rate. But if there are repeated sequences, then you can go for the blue one here, and you have a green one, but it might cross over like that. So should have been here. And here's a repeated sequence. Oops. So you get unequal crossover. So what happens now, it's like that, we get one little tiny short one here, and one much longer one. Okay. So this is what you might call unequal. This is called unequal crossover. And when you think about it, this is a very natural bug. It's not at all clear how cells avoid this. It's horrendous. How bad is that? So now you've crossed over in the wrong place because there was a repeated sequence. About a fifth of our genome comes from a genetic parasite. Those are repeated sequences. About 20% of the human genome. Yep. So there's a lot of repeated sequences, which very slowly accumulate mutations. Repeated sequences, you can get this crossover. So the point is, now the next thing I'd ask you is which one of these is more likely to survive? Sorry, can you all see what I drew there? Oh dear. I'll draw the unequal crossover again up here. As Mark said, you have a little short one, or a crossover. You cross over in the wrong place, and you can have a great big long one. Okay, so these go into a separate and their gametes, or eggs or sperm cells or whatever, which one of these is likely to be better? Yeah.
The longer one.
The longer one. Yes. Why?
It has more variability for the different changes in the environment. Given that the cost is such that it can sustain it.
Why does it have more variability?
Because it took long sequences of more species.
Yeah. The reason is the long one has got everything with some bits of it twice. Having something twice is generally not as bad as not having it at all. So this thing is missing large numbers of genes. This one has got some extra genes, it's got some genes duplicated. This may or may not be good, possibly not good, but it's much more likely to be okay than missing something. If you've missed out a few 100,000 bases. This is going to work better. And unequal crossover is not at all a rare event, because everyone knows what a crossover by. This is an aspect of sexual reproduction which is pretty hazardous. You're cutting up your blueprints and stitching them back together. If you make a mistake, you got this. So there's a tendency to increase genome size. There's a natural tendency to increase the genome size by unequal crossover. And so this is one reason why that graph which I showed, some lineages have accumulated absolutely enormous genomes and huge variation in genome size. You can also get polyploidy, where you have a thing where you manage to have twice as much genome as you should have had. You manage to get two complete nucleuses, or a nucleus containing two complete genomes. But that only gives you a factor of 2. It's the continuous unequal crossover that gives you huge growth in genome size. It's rather difficult to select for chopping bits out. So all this huge genomes can arise through this chance process. What about the classic GA one-point crossover? Have you looked at? Did you do one-point crossover genetic algorithms?
So.
You think so? Mark must have shown you these things. Sorry?
Yeah, the basic crossover.
The basic crossover method. So, is this a good and necessary model? Well, maybe not. There is a whole class, a very successful class of organisms you can find in every pond. If you look outside and you look at sort of puddles and wet bit in the park outside, you'll find plenty of protozoans or ciliates, and they have a really weird genetic mechanism. Let's look at some ciliates. If you go to a pond, you can find stentor, which sits there. It's actually large enough. This is a single cell which is big enough to see. Just to give you an idea of the variety of life. The record size for a protozoan cell is like 5 centimeters across in the deep sea. These you can just about, they're about 1 millimeter long, and they sit there sort of hoovering up something. It's a single cell. It doesn't have a nervous system. It actually has some adaptive learning. The is how, as far as I know. The remarkable thing about it, here's parameciums. By microscopes as children, see parameciums hoovering around, all like sort of sharks. Only they're like 0.5 millimeter long. They have a really weird reproductive method. What they have is they have 2 nucleuses in the normal cell. They have a micronucleus, which is that, if you like, contains the DNA that gets inherited, and a macronucleus, which contains the DNA that gets used. So the micronucleus gets inherited with sort of not very many points of crossover, like pretty much everything, but the macronucleus is formed from the micronucleus. I don't want to belabor this point too much. I enjoy a very beautiful diagram. Basically, they chop up these segments, the micronucleus segments, into many hundreds or thousands of small lengths, and it all gets reassembled in a different order. And with hundreds of copies put into the macronucleus, whereas this is where it gets used, because it's a huge cell, so it needs loads and loads of copies. It needs lots of DNA. People believe that it has slightly higher rates of. There are articles suggesting that ciliates have higher rates of protein evolution as a result of this process. have no idea if it's true or not. But it doesn't look like one-point crossover to me. It's a one-point crossover, but in a very different style of genetic representation, which avoids most of the alleged motivations for one-point crossover. So a single-point crossover doesn't seem terribly essential. Different animals have wildly different numbers of chromosomes. So that's an extreme case. Another thing, diploid or haploid. Are you happy if you're a diploid, you've got 2 copies of each chromosome. If you're a haploid, you've got only 1. And most complex organisms or animals are diploids. Actually, no, most lineages are diploid, and you might say, oh, well, this is, I know have been people, I'm mentioning this because in the genetic algorithms literature, there are people who say, oh, let's use diploid genetic algorithms. That will give us more biological reasonableness. But actually, when you look at all sexual lineages, have a haploid phase and a diploid phase of their life cycle. So you're in a diploid, we are all in the diploid phase. Our haploid phase is generally brief and small. Of course, eggs last for a long time, sperm less so, eggs and sperm. So there's meiosis, with this crossover of these 2 chromosomes, which leads to the gametes here. 2 gametes, usually gametes from a different organism, they join together to form a new diploid with syngamy, and this develops into an organism. That's clean. So it seems, oh, well, maybe, if we think about animals, maybe you need to be diploid to be complex. Well, not really, because there are some orders of plants, mosses and liverworts, where the diploid phase is tiny and very brief, and the actual complex organism is entirely haploid. Some insects are haploid-diploid, and that the males are haploids and the females are diploids. Conclusion, you don't really need a diploid genome. If I was doing GA research, I would start. These are some pictures of mosses and liverworts. They're sort of small but reasonably complex and highly visible plants now. Hundreds of species, or thousands of species, but certain times in the geological past, mosses have been about 30 feet tall, so they can get big. So haploids, there's nothing, there isn't so much wrong with haploids. Let's have some terminology. Loci and alleles. So, in biological terminology, each position on your genome, this is a thoroughly imprecise biological term. If I start trying to define a locus mathematically, I'll probably fail. But let's just try. Let's just roll with this. The locus is a section of genome that's basically inherited as a unit and doesn't normally get split up. You say, oh, hey, excuse me, any place on my genome can be split up by crossover. Well, yes, that's true. But when biologists talk about a locus, they are usually talking about something hundreds or thousands of bases long. The reason is that in humans, crossover happens about every 100,000 bases. So there are bits of our genome that haven't recombined since before we were human. So a locus is a section of genome that's basically inherited as a unit and it's not recombined with crossover. So let's just suppose that the genome consists of these beads, little blocks which do not recombine. They're called loci. The locus is loci. Now the sequence out of locus can mutate. So different individuals may have different versions of this genetic sequence. Each particular version is called an allele. Don't ask me why. Don't ask me why, because I should know and I don't. You can probably find from Wikipedia derivations of what allele is. So, the next question is the next thing about sexual reproduction is that every child has 2 parents. Let's think about this. Is this an essential thing or an accident? I mean, in nature, biologists have long regarded sex as a huge puzzle. That is, it's dangerous, it's risky, you've got to do crossover, you can catch things, there's all sorts of, there seems to be a great deal of extra mechanism involved. When parthenogenesis, budding off, is just so much simpler. So why does it exist? But all sex in nature recombines two genomes. I won't go into it, but if you want to do mathematical analysis, you have a population of genomes, and then your next population consists of children, each of which has two parents. This is really hard to analyze. It would be much nicer to be able to say, okay, let me draw this at a level at which you can see it. Let's do a mathematically much simpler idea. It's the following. Supposing I have my genome, I'm going to have arrays as genomes. So I'm going to have these little beads, and they're going to be, so let's put the things on zeros and ones. They might not be. And so on, coming down here. And if I want to breed something, I could pick two of these and recombine them, or alternatively, I could produce a new one by, say, eliminating one of the old ones, and I randomly pick an allele from any member of the population. So I have a weird kind of, if you like, group sex going on here. So I want to call this n-way crossover. Sometimes people call it n-way crossover. There are 20 names for it. It's the first simplifying assumption people make in population genetics. It's the we'll come back to it. But this is a rather nice idea, because now I'm selecting each of these things here independently. I can select each of these elements independently from any member of the population. And this looks kind of like sex, only not. You might call it kind of ultra-sex or hypersex or something. So people have tried genetic algorithms doing this, and I'm not aware of any significant results where they show that this is any worse. And indeed, it's rather frequently better on the genetic algorithms problems that people try on. So this seems, the reason this is nice is if we have just zeros and ones, we don't have to actually implement this as n-way crossover. We don't have to literally do this process. We can just keep track of how many zeros and how many ones there are in each column. And so we can take up, if you like, the marginal distribution. And here we'd say it's, I know, it's 1.0 is the fraction of ones. Here it's 0.0 is the fraction of ones. And here it's, let's say 0.9. And this one we've got ones and zeros. So it's maybe it's 0.7. This one will also be 1. That's going to be 1.0. And here it's all 0.0 and so on. And then we can just, for a new element, we can select a 1 or a 0 with these probabilities. So what we're doing is we can maintain a marginal distribution, maintain a concise estimated distribution. We will come back to this, don't worry. As a representation of our population, so you don't actually have to maintain the whole population. Every time you generate a genome, you have to consider its fitness, but you don't have to do this crossover process. Now, again, I just want to pause to highlight this modeling assumption for two reasons. The first is to say that this is quite a major assumption. Here I'm not saying a piece of complicated biology is irrelevant. I'm saying let's make an outrageous mathematical simplification and hope that it works. So this is of a different kind. Actually, it's not so outrageous. Let's tell a just so story. Supposing I have a population and I'm considering the evolution of something which is really useful during, I don't know, some disaster, a drought that only happens every 100 generations. Nevertheless, so in that time, so we only have selection every 100 generations instead of every generation. Still have evolution for this. Maybe useful at that rate. But in that case, there's a lot of time for every time we select, the individuals we're selecting on are combinations of the genomes of the ancestors who were selected, but there have been 100 generations of jumbling up. So it's quite similar to this. Yeah. So this is a simpler type of. So the question is I'm just raising this question simply to note that I'm going to make this assumption. Because even if you think that two-parent reproduction really is important, I'm not going to analyze it. There have been some people who tried. Oh, I'm sorry, this slide. I hate Microsoft Word so much. What I mean is this. Dear. I'm terribly sorry. Choose them. I'll edit argument so far. Where am I? Here, 2. What do I do? Control-Shift plus. To the number of differences. And here I have 2. Control-Shift plus. Hash. So what I'm trying to do is to model, and when we model, in modeling you can do whatever you like, but it's a good idea to note when you're doing something naughty. Note when you're making what assumptions you're making will go on. Here I want to note that it's a fundamental thing about sex, for possibly only for mechanical reasons, that you only get reproduction from 2 parents. I mean, how do you arrange it in nature? Tricky. But I guess if you had, octoploids, then you could have 8 parents. That sounds good. It'd be tricky to arrange. All sorts of things could go wrong. Sexual reproduction has enough flaws in it. So the difference, there's a real combinatorial difference. You've just got, say, each offspring is the product of only 2 parents. Then the number of different possible offspring you can produce is very limited. With 1 point crossover, hey, you can only choose 1 point crossover along the gene, along the genome. So the number of different offspring you can produce is actually only the length of the genome. If your genome is length 100, there's only, and you have 2 genomes there, which are going to combine, there are only 100 possible offspring you can produce. It's a very small number. If you have uniform crossover, I trust that Mark Herbster has explained uniform crossover, where basically you take 2 genomes and you pick one element from one, and you say, oh, I'll take the next element from this one. You toss a coin and you pick the next element from whichever genome, so you do crossover all along the genome, in many, many, many places, randomly. If you do that, then still there are not that many different children you can produce, because in many places those 2 genomes will be similar, will be the same. And so you cannot produce a difference. You can only produce different children where there is a difference between the 2 parents. With n-parent crossover, wait a moment, in 1 generation you can produce a hugely greater number of different possible children, because this time, if we're sticking to 0s and 1s, there will be certain places in the population where, hey, everybody's got a 1 there, you won't have 1 there, everyone's got 1, everyone's got a 0 there, yeah, you better have that. But these are polymorphic here, where some of them have got 1s and some of them have got 0s. So if everywhere there's a polymorphic locus, poly means many, morphic means type, with many types of element in it, then you can produce, you have choices as to which thing you put in, and they can all come from different parents. So I'm not going to slide this past you. Now, what I think is interesting, and again, if any of you are feeling like procrastinating from your other work, or are sufficiently interested to follow this up, I would be most grateful, is that I don't know of any anyone who has asked the obvious. This is a very common approximation. It's a very common assumption made. I don't know of anyone who has shown that algorithms with just two parents, genetic algorithms with two parents, are more effective than these. In fact, these actually have usually done better. There's a well-known paper from about 1995 by a man called Shumit Baluja. That's his title, Mark Baluja. He was a very empirical guy, and he wrote a paper which he submitted, I think it's in a machine learning conference called Taking the Genetics Out of the Standard Genetic Algorithm, where he recommended doing precisely this. And he was a very empirical guy, and he took a lot of benchmark problems, and he found that for most of them, his method, for them, they crossover, did slightly better. I don't want to claim it's better, I don't want to claim it's worse. What I'm saying is that two-parent reproduction is an accident. That's an assumption. It may not be true. So the argument so far, first, is that evolution is highly effective. People say that evolution is very slow. I've shown you the meet the mothers and the sperm whales. I'd like to suggest it's very effective. I mean, I was talking to Chris Bishop at Microsoft Labs at a conference. He was possibly jet lagged at breakfast, and possibly he didn't want to talk to me. I was bending his ear about evolution. And eventually he said, Chris, what people don't realize is that evolution takes place on a time scale on which granite is a liquid. It's long enough for things to happen. Well, I should have retorted to him, but I did not, that it is very curious to hear a computer scientist appealing to brute force. Brute force in algorithms is usually a bad idea. And I hope that the examples of the whales and of meeting our mothers will convince you that there is something startlingly effective about evolution, which stands in need of explanation. And in the world, organisms haven't even got it, startlingly different life cycles and lifestyles, genome sizes, we've seen, genetic architectures, think of those ciliates, population sizes. There are organisms. What is very weird when you think about it is that we think that we're pretty much the most complicated organism on the planet. I mean, be honest. Do you really think that anything else is much more complicated than you? If you're feeling particularly self-critical and unself-confident, you consider dolphins are much more intelligent than me. Yeah, really? Not really. So we're we're up there with dolphins. Now, us, dolphins, and elephants, and whales, and other interesting creatures, we think, probably, and parrots, okay, crows, we think that we are the most complex products of evolution, the most marvelous products of evolution, and yet, we have a tiny population compared to bacteria and protozoans, and insects and flies and aphids and mosquitoes, and malarial parasites, which are little multicellular sexual things. And we reproduce the most slowly. So we've got the smallest population, and we breed the most slowly with the fewest children. How is that? That's kind of remarkable, right? You would think, if you could breed like crazy, and you had a huge population, you could do better. Well, numbers of offspring. I mean, cod have 1 million, we have 5. Generation time. So there are not very many universal characteristics of life. Evolution seems to be a remarkably robust process which does not require special conditions in which to succeed. As far as we know, there is nothing kind of inherent. Yeah.
Sorry. I was just going to say, with regards to the n-way crossover, is there not a fundamental constraint with that? Because this model sort of assumes encapsulation of each element, and
yes.
you can flip it, but with peptides and proteins, you need a very specific sequence of lots of proteins to achieve. Of lots of peptides to achieve.
Nice point. Yes. Okay. For any particular protein. Okay. So the biologist's locus is something a few hundred bases long, right? 10 or. For a biologist, what they mean by locus, a place on the genome, is going to be like a protein, or a protein domain, or something, or a binding site, or whatever. Anything between 8 to 800 bases. Yes, so drawing this as ones and zeros, I don't mean to imply these are individual DNA elements. I mean to imply they're individual loci. And I just draw them as ones and zeros to make the connection with genetic algorithms, to give you the contrast between one-point crossover.
Even if they were loci, would that overcome those constraints?
Well, if they're loci, this is, right. What I'm saying here is that you can have an offspring which is that there are going to be no correlations between what loci, what alleles are at what positions. Okay? And so we if I know what this locus is, what allele I've got with this locus, it doesn't tell me anything about that locus. Whereas if you've got a breeding population, you might have individuals which are fit if they've got an A sequence peptide here and a C sequence peptide there, or alternatively they've got a B sequence peptide here and a Z sequence peptide there, and nothing else will do. But then, if you have very small amounts of crossover, you can maintain these correlations more easily. So there is a difference, as you point out. So
where would you recommend learning more about these ideas?
Right, there is a book which I intended to bring with me. I would not recommend picking up any random. I'm sorry, I will put it on. I'm sorry, I intended to bring a book with me to show you, which is a slim volume written in an elegant way, from which I have taken much of my discussion of population genetics. And I find that an elegant and comprehensible book written by somebody sensible, as opposed to someone who is obsessed with detail. And should I remember the rice? Yeah, I think it's. But I will bring it next time. Or I'll be putting things on Moodle when I've got access to UCL computers again.
Thank you.
So that's it. And Sean Rice. The author is Sean Rice. Published in the early 2000s, and it's his book. And so if you Google Sean Rice and genetics, you'll get that. And personally, it's a slim volume, it's not too long. He takes elegant approaches, and I think he concentrates on a lot of the right things. And the problem with most, again, don't think I'm making a kind of blanket condemnation of a lot of genetics books, but what they do is they will do something elegant. They will say, I'm going to consider one locus and haploids and what happens to the distribution of alleles at one locus. And then, damn, the next thing they do in the next page, they say, well, right, we're going to apply all our formulae to diploids. It gives you factors of 2 all over the place. It's dreadful. Everything is expanded. And then they consider all the other special cases that you get to in biology without sticking to the essentials. And Sean Rice sticks to the elegant essentials for as long as possible. in a short and elegant book, as opposed to a vast and over-detailed book, which is just full of inelegance, which comes from considering diploids and linkage and all sorts of things. So, what did we so far? So I will put this up. So the universal characteristics. Sex, the same genetic, I'm not going to touch on the genetic code, but the genetic code is a universal characteristic. Maybe it's a universal accident, maybe it's particularly good. Certain developmental mechanisms, as in paper 2 that I gave you, seem very highly conserved. I mean, it's a valid question for life on Earth. Is the effectiveness of evolution, of the structural evolution of multicelled creatures, the result of evolving particular developmental mechanisms by some early chance, which are then highly conserved, which allow massive proliferation of structural complexity in it. And so we're going to do that. But the first thing, before we get to the complicated, we're going to study the tiniest genetic algorithm possible. When in doubt, study something absolutely as simple as possible and understand it completely. This is what I'm going to do next week. We're going to have genomes, we're going to have one locus. Our organisms are going to be ones or zeros, or A's or B's. We're going to have a population of n of them, and we're going to be reproducing them one at a time, and we're going to say that one of them is, well, a 1 is slightly better than a 0. Obviously, it could be the other way around. And if we couldn't understand this model, then we can't understand much. Actually, if you look in the books on evolutionary computation, it does seem reasonable, I think, to ask questions such as, is it necessary to have two parents or not? Does it really make a difference? And secondly, what does this model do? They don't ask us. So we won't. And the great thing is, luckily, we can see that we can understand this model precisely. We can just lay it out onto the table, simulate it in MATLAB, and draw the theoretical curves and look at its agreement and know exactly what happens. Then we can put multiple copies of this model side by side and have larger scale genetic algorithms. But it's cool to understand this. Notice that there is actually a lot to understand here, because there are 3 parameters. Ouch! There is the population size. It's a finite population. There's the mutation rate, which I forgot to mention on the slide. And then there is S, fitness. That's a lot. It's awful. Even in the very, very simplest model we have, there are going to be 3 parameters, and we're going to understand how they interact. Luckily, they interact fairly simply. And it turns out to be, to cut a story short, it turns out that this is thoroughly connected to Bayesian inference. The model for this is exactly a Collier-Earn model. If we're allowed unlimited alleles, we get a Dirichlet process. And you may have encountered in reading in machine learning something called the Chinese restaurant process, and looked at this in some trepidation, especially when it's followed by the Chinese restaurant franchise. This was originally invented, or one of the distributions was originally invented by mathematical geneticists, so what I'm trying to do is actually return genetic algorithms to their mathematical genetic roots. But it's great. We can have a simple process, just one set of zeros and ones, understand it totally, precisely, plot it out in MATLAB, and then stick it together into something complicated. That's what we're going to do next week. And this will also not be gratuitous maths, unrelated to other aspects of your course. What I'm going to end up with is an explicit representation of the equilibrium distribution of this genetic algorithm as a Bayes net in the form of a factor graph. We've done Bayes nets in the form of factor graphs, as it has been mentioned. Good. We're going to do more. That's good. We can do some more. Okay. So it does. So the other advantage of doing it this way is it will relate to the rest of the course. Okay. You've been remarkably patient sitting here for 3 hours while it gets dark. And I will try to get access to Moodle as soon as possible.